Good evening. Welcome to session 10 of the NPTEL course, Genome Editing and Engineering, which is taken by Professor Utpal Bora of IIT Guwahati. I'm Sanket from the School of Biological Sciences, IIT Delhi. We will be beginning in a couple of minutes. Let's wait for everybody to join in and then we can start. Okay, so let's begin. The question one is the process of spreading to other organs is called DASH and is a major cause of death from cancer. Is it A, mutation, B, metastasizing, C, tumorigenesis, or D, neoplasm? So I'll give you a minute to think on this, and then we'll discuss the answer. Okay, so the process of spreading of cancer to other organs is called metastasis. So if you look at how cancer progresses, it's usually a normal cell which divides, as you can see on the panel one. So it performs its function. When there is a mutation, essentially not a single mutation, but an accumulation of mutation, that mutated cell divides uncontrollably. That leads to cell proliferation and increase in the number of these mutated cells. These mutated cells eventually form a mass of cells or tissue, and that is what we call as tumor. These tumors can then flow through the blood vessels to different parts of the body, and this very process is called metastasis. And when this tumor it breaks loose to other tissues and organs, we call it as malignant tumor. Otherwise, it's a benign tumor. Question 2. M1 macrophages are able to 1. Release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-1 and IL-23, and INOS. 2. Re-educate the DC and CD4 plus T cells. 3. Deactivate CD8 plus T cells and as a result promote an immune response against the tumor and prevent tumor progression. Choose the correct options. Is it 1 and 3, 2 and 3, 1 and 2, or all of the above? Okay, so when we talk about T cell exhaustion and differentiation in TME, T cell exhaustion is a hyper-responsive state of T cells in chronic environment with increased inhibitory receptors, decreased effector cytokines, and impaired cytotoxicity. Most T cells in tumor microenvironment are exhausted, leading to cancer immune evasion. Native T cells, CD44 low and CD62L high, activate and differentiate into effector T cells in secondary lymphoid organs, as you can see on the figure in the right? When effector T cells enter the tumor microenvironment, they are polarized into exhausted T cells with decrease in effector cytokines and increase in inhibitory receptors. Subsequent exhausted T cells may turn to be defective memory T cells or be deleted physically. Stimulants present in the tumor microenvironment such as cytokines, chemokines, and growth factors determine the polarization of macrophages and their differentiation towards M1 or M2 subtypes. M1 macrophages are able to release pro-inflammatory cytokines such as IL-12, IFN gamma, IL-1, IL-23, and INOS. They can re-educate the DC and CD4 plus T cells, and they can activate CD8 plus T cells, and as a result, promote an immune response against the tumor and prevent tumor progression. In contrast, M2 macrophages and tumor-associated macrophages increase angiogenesis and formation of tumor-associated fibroblast. These cells attenuate immune response in the tumor microenvironment and increase tumor progression. The state of tumor microenvironment strongly affects the patient's prognosis. Identifying the mechanisms underlying the tumorigenic characteristics of interactions between immune suppressive cells and tumors can reveal novel therapeutic targets for developing antagonists such as monoclonal antibodies and immunomodulatory drug interventions. Another approach is to fortify already existing immune responses to or develop new ones through bypassing their dependence on the robust and intact immune system, which becomes non-functional in a tumor microenvironment. Immunotherapy of cancer has recently proven its potential in numerous clinical trials. Question 3. Immune checkpoint inhibitors are designed to reinstate anti-tumor immune response by interrupting co-inhibitory signal pathways and to promote immune-mediated elimination of malignant cell, immune cell, tissue cells, or cytokines. Obviously. The answer would be malignant cells. So T cells in peripheral blood infiltrate into the tumor and undergo functional state transitions, possibly driven by immunosuppressive microenvironment. Native CD8 T cells or CD4Th helper cells differentiate into transitional states and finally reach exhausted state while resting T regulatory state cells. They transit into suppressive states in tumors. Such state transitions result in a reduction of effector T cells, yet an accumulation of exhausted T cells and suppressive T regulatory cells, both of which are proven to be proliferating and highly clonally expanded in the TME. Myeloid cells in blood are mainly monocytes, including CD14 plus and 16 plus subsets, while these cells tend to differentiate into macrophages and dendritic cells in tumors. The tumor microenvironment sculpts them to harbor immunosuppressive phenotypes, resulting in an accumulation of suppressive TAMs, tumor-associated macrophages, and CD2s, this is classical dendritic cells, but a reduction of CD16 monocytes and CDC1s. In addition, single-cell interrogation facilitates the identification of novel subsets of classical dendritic cell and TAMs in the TME and reveal that TAM subtypes tend to co-express M1 and M2 signatures, thus inconsistent with the polarization models. Natural killer cells, they exert cytotoxic functions with perforin and granzymes when activated by integrated signals of activating and inhibitory receptors. Yet they show reduced cell numbers, impaired cytotoxic function, and an impeded orchestrating effect for immune responses, exemplified by the hampered CDC plus classical dendritic cell recruitment in the TME. The functional defects of natural killer cells are possibly driven by tumor cells through secreting immunosuppressive factors and expressing ligands of inhibitory receptors, while decreasing the expression of ligands of activating receptors to hinder natural killer cell activation. These cells, they play an important role in anti-tumor immunity and immune checkpoint inhibitor treatment, as B cells and tertiary lymphatic structures containing aggregates of immune cell including T cells, B cells and follicular dendritic cells are found to be to mediate improved responses to the ICI or immune checkpoint inhibitors, the mechanism of which involves activation of TFH in B cells. The activated B cell can differentiate not only into plasma B cells to produce antibodies to clear cancer cells, but also into active T cell mediated immune response representing antigens to CD4 T helper cells that could promote the activation of CD8 T. Question 4. Cancer vaccine takes advantage of tumor-associated antigens or tumor-specific antigens to stimulate the immune response, especially a robust and long-lasting immune response of CD8 plus T cells to inhibit dash of tumor cells, growth, infection and recurrence, killing metastasis and recurrence, growth, metastasis and immunosuppression, or growth, metastasis and recurrence. Cancer cells, they take advantage of tumor-associated antigens or tumor-specific antigens to stimulate the immune system, especially a robust and long-lasting immune response of CD8 plus T cells to inhibit the growth, metastasis, and recurrence of tumor cells. That is option D. According to the clinical use of cancer vaccines, they are divided into two categories. Either they are preventive or therapeutic. Preventive cancer vaccines, it aims to prevent the occurrence of a tumor by inducing immune response, while therapeutic cancer vaccines are designed to eradicate tumor cells by inducing or enhancing the tumor specific immunoreactions. So cancer vaccines, regarding that, what we know is that the efficacy is primarily dependent on the immunogenicity, host immunosuppression, preferential expression of tumor antigen, and the delivery of cancer vaccine. The tumor-specific antigens, also known as neoantigens, are ideal cancer vaccine targets due to the highly immunogenic, lower risk of self-tolerance, less common immune tumor antigen deletions, and lower risk of autoimmune reactions. So far in phase 3 randomized trials, there is no other therapeutic cancer vaccine yet which has shown noteworthy clinical efficacy except the Spulucil T. The heavy chain variable region and light chain variable region of antibodies are linked by a small segment of polypeptide, CAR T cells, tumor cells, or CD3 zeta chain. As you can see here, there is a chain of, there is basically a chimeric antigen receptor, how antibodies usually look like. So it is, it's composed of a membrane distal single chain variable fragment, SCFV, a spacer domain, a transmembrane domain, and an intercellular signaling transactivation domain. In contrast to TCR, CARs recognize surface antigens in a non-MHC manner. TCR identifies intracellular or extracellular proteins that are presented as peptides by MHC molecules. CAR T cells are typically produced by transducing T lymphocytes with a transgene encoding a synthetic antigen. This transgene is integrated into the T cell genome, transcribed and translated into CAR protein or chimeric antigen receptor protein. The binding and signaling domains are separated into extracellular and intracellular compartments respectively. The extracellular SCFV domain is derived from the variable regions of an antibody that recognizes specific tumor antigens together with a spacer that provides flexibility in the binding domain. The transmembrane domain connects the binding domain with intracellular signaling moieties, whereas the TCR derived CD3 zeta chain drives T cell activation and is fused in tandem with co-stimulatory endodomains that allow for robust and sustained function. Extracellular single chain variable fragments work for antibody-like antigen recognition and intracellular signaling domains for activating T cells. In CAR T cells, the extracellular domain is CFV is responsible for redirecting the specificity of CTLs to the malignant cells or cytotoxic T lymphocytes to the malignant cells, and can be designed according to the specific antigens such as CD19 expressed in B cell acute lymphocyte leukemia, chronic lymphocyte leukemia, and lymphoma. CAR intracellular signaling domains provide the necessary signals for priming T cell activation. In the CAR T cell mediated immune response, the SCF of chimeric antigen receptors can engage surface antigens of tumor directly via antibody-like binding. So the heavy chain variable region and the light chain variable region of antibodies are linked by a small segment of polypeptide. The hinge domain is composed of immunoglobulin superfamilies such as CD8, CD28, or IgG, which play a role in signal transduction. The intracellular signal transduction region is mainly composed of CD3 zeta chain of the T cell receptor. In addition to intracellular signaling domains, co-stimulatory molecules such as the CD28 or 41BB can improve cell proliferation and survival time in vivo and enhance the anti-tumor activity of CAR T cells. When CAR T cells bind to tumor surface antigen, they proliferate and kill tumor cells. Well, question 6. Toxic levels of systemic cytokine release and severe immune cell cross-activation in some patients result in the following toxicities: cytokine release syndrome, hemophagocytic lymphohistiocytosis, macrophage activation syndrome, immune effector cell associated neurotoxicity syndrome. Which of the following option is correct? Is it 1, 2 and 3, 1 and 4, or all of the above? So the toxicities underlying, wait, just give me a moment. Yeah, the toxicities underlying CAR T cell therapy have been most extensively characterized in patients in the first FDA approved CAR T cell therapy, CD19 directed chimeric antigen receptors. 23 to 46% of patients displayed severe supraphysiologic cytokine production and massive in vivo T cell expansion. These toxic levels of systemic cytokine release and severe immune cell cross-activation in some patients result in the following toxicities: cytokine release syndrome, that is associated with supraphysiologic cytokine production and massive in vivo T cell expansion; hemophagocytic lymphohistiocytosis or macrophage activation syndrome, that is a severe hyperinflammatory syndrome characterized by cytokine release syndrome and combinations of elevated serum ferritin and hemophagocytosis, renal failure, liver enzymes, splenomegaly, pulmonary edema, and or absence of natural killer cell activity; immune effector cell associated neurotoxicity syndrome, that is elevated cerebrospinal fluid cytokine levels and blood-brain barrier disruption. To overcome the barriers of limiting wide application of CAR T cell therapy, multiple strategies have been developed. One of the most feasible and durable approaches is to generate allogenic universal CAR T cells from healthy donors. Compared with autologous CAR T cells off the shelf, allogenic CAR T cells have many potential advantages, including immediate availability of cryopreserved CAR T cells for patients in urgent need, enough quantity for first infusion or redosing, and possible standardization of CAR T cell producing. Whereas, considering the presence of endogenous HLA or TCR on donors' T lymphocyte, the biggest challenge of universal products is the potential risk of alloreactivity, host versus graft response, and graft versus host disease. Genome editing has been widely used to generate off-the-shelf allogenic CAR T cells, and that removes alloreactivity and to address other limitations associated with immunotherapy strategies like TCR T cell therapy. Question 7. In an application of CRISPR in which the effect of T cell function on cancer immunotherapy in the presence or absence of a gene is investigated, and the results may use for the development of further immunotherapeutic approaches is known as loss of function screening, knocking out granulocyte macrophage colony stimulating factor, immunosuppressive tumor microenvironment, or CAR screening. So loss of function screening is another application of CRISPR in which effect of T cell function on cancer immunotherapy in presence or absence of a gene is investigated, and the results may be used for development of further immunotherapeutics. By adopting such study, one group by Schifrud revealed that the knockout of some T cell regulatory proteins such as the SOCS1, TCB2, RASA2, and CBLB improves both proliferation and anti-cancer capability of modified T cells. To elucidate immunomodulatory impacts of combinatorial role of anti-cancer on cytotoxic properties of affected T cells, and to explore the mechanism of CAR T cell cytotoxicity, a detailed in vitro loss of function screening by means of CRISPR was conducted recently. The first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing TCR T cells in patients with refractory cancer was reported in A 2019, 2020, 21, or 22. So about clinical trials, there were several anti-CD19 CAR T cell constructs that have been investigated, and the response they differ extensively among various studies in patients with hematological malignancies, especially refractory or relapsed acute lymphocytic leukemia. In 2017, the Food and Drug Administration granted regular approval to axicabtagene, ciloleucel, or Yescarta as a therapeutic option to treat pediatric or young adult B lymphoblastic leukemia, an adult diffuse large B cell lymphoma. Yescarta is a CD19 specific CAR T cell mainly exploited for the treatment of adult patients with relapsed or refractory large BCL following two or more lines of systemic treatment. In July 2020, Tecartus was approved for the treatment of adult patients with mantle cell lymphoma. In February 2021, Breyanzi, the fourth CAR T cell therapy for adults with relapsed and refractory large B-cell lymphoma, was approved by US FDA. CAR T cell therapy is more restricted in solid tumors than in hematological malignancies, as CAR T cells are circulated to the bloodstream and lymphatic system and thereby have more interaction with blood tumor cells. The first clinical trial testing the safety and feasibility of CRISPR-Cas9 editing TCR T cells in patients with refractory cancer was reported in 2020. The safety and efficacy of CRISPR-Cas9 edited universal CAR T cells in vivo needs to be further tested in clinical studies. Currently, 8 relevant clinical trials are going on, which is already given in the table. Which of the following is not a correct match for surface antigen targets for CAR T therapy against their cancer types used for clinical trials? Is it carcinoembryonic antigen for colorectal adenocarcinoma, fibroblast activation protein for malignant pleural mesothelioma, diganglioside GD2, neuroblastoma, glioblastoma, melanoma, and osteosarcoma, human epidermal growth factor receptor 2, HER2, for colorectal adenocarcinoma. So option D is the answer. Question 10. What is cytokine release syndrome? Is it the severe hyperinflammatory syndrome characterized by combinations of elevated serum ferritin and hemophagocytosis and renal failure? Is it B. Severe hyperinflammatory syndrome characterized by combinations liver enzymes, splenomegaly, pulmonary edema, and or absence of natural killer cell activity? Is it C. Associated with supraphysiologic cytokine production and massive in vivo T cell expansion? Or is it D. Elevated cerebrospinal fluid cytokine levels and blood-brain barrier disruption? Answer is C, that cytokine release syndrome is associated with supraphysiologic cytokine production of cytokines in large numbers, and massive in vivo T cell expansion. Which TCR complex on TCR engineered T cells is responsible for anti-tumor activity? Is it the endogenous T cell receptors, the transgenic TG TCR containing transgenic alpha and beta TCR subunits, combinations of TG and endogenous subunits as mixed TCA, or none of the above? Would be the transgenic TG TCR containing transgenic alpha and beta TCR subunits. Who is often called the father of cancer immunotherapy? Is it Paul Ehrlich, Dr. William Coley, Lewis Thomas, or Sir Frank McFarlane Burnett? It's option B, Dr. William Coley. Which of the following is not a feature required for a candidate target of antigen that are used for TCR engineered T cell treatment? Is it they must be selectively expressed in tumors and not in normal tissues, that is the tumor specificity? They're related to oncogenesis, tumor addiction. They're able to evoke a T cell response, immunogenicity, or they must be selectively expressed in both tumors and normal tissues, or tumor specificity. So the answer is they must be selectively expressed in both tumors and normal tissues. Which of the following correctly defines a tumor-associated antigen? So tumor-associated antigens are peptides that originate from endogenous wild-type proteins. Their expression is elevated in tumors, limited in magnitude or in spatial expression in healthy tissues, or all of the above. Okay, so the answer to this would be all of the above. Question 15, which of the following is one of the major methods of adoptive cell therapy? Is it use of cancer vaccine, use of TCR, T cell receptors modified T cells, use of cytokine interferon alpha, use of immune checkpoint inhibitors? Use of TCR, T cell receptors modified T cells. Genome editing technologies such as zinc finger nucleases, transcription activator like effector nucleases, and CRISPR-Cas9 can be applied to knock out the T cell receptor alpha constant and T cell receptor beta constant loci to prevent A. Ability of CAR T cells to kill tumor cells, depletion of programmed cell death protein 1, tumor infiltration by TIL, or alloreactivity due to endogenous TCRs? The answer is alloreactivity due to endogenous TCRs. So to prevent that, we go for these genome editing technologies. What is the role of TCR and co-stimulatory signaling domains in CARs, or chimeric antigen receptors? Is it to enhance the binding specificity of antibodies, to enable direct recognition of major histocompatibility molecules? They activate T cells or prevent the activation of T cells? Answer would be to activate T cells. And the last question for today, which of the following options correctly describe the enormous potential of CRISPR-Cas9 techniques in cancer detection? 1. CRISPR-Cas9 based diagnostic systems Sherlock, that is specific high sensitivity enzymatic reporter unlocking, and detector providing T cell receptor knockout, TCR knockout, chimeric antigen receptor T cells, CAR T cells, elimination of oncogenic virus like HPV, and establishment of in vivo tumor models by eliciting mutations in several genes. Knock-in of inhibitory receptors such as PD1, programmed cell death protein 1 and LAC3. Is it 1 and 2, 1, 2 and 3, 1, 2 and 4, or all of the above? So the answer is 1, 2 and 3. So CRISPR-Cas9 based diagnostic systems can be used for Sherlock and detector to provide T cell receptor knockout, chimeric antigen receptor T cells, and elimination of oncogenic virus like particles. For establishment of in vivo tumor models and knockout of inhibitory receptors, these are not essentially the potential of CRISPR-Cas9. So with this, we are done with the today's session, and I hope you guys liked it, and I'll just stop the recording at this point, and if there are any questions from your end, now would be the time to ask.